I'm Rick Lifton, the president of Rockefeller University, and I'm delighted to welcome you to this evening's program. It's a pleasure to see so many of Rockefeller's benefactors and friends here, and to see new faces in the audience as well. For those of you who are new to Rockefeller, I'd like to say a few words about the university. We were founded in 1901 by John D. Rockefeller, who recognized that the major limit to advancing the diagnosis and treatment and prevention of disease was a lack of understanding of the fundamental processes that underlie normal life functions and how they go awry in disease processes. Rockefeller was created to bridge this gap, establishing our credo, science for the benefit of humanity, which has fueled our efforts over the last 117 years. We are small by traditional university standards. We have only 82 faculty laboratories, each of which is led by a remarkable scientist who reports to the president, and all are broadly engaged in the study of life sciences. We recruit scientists who are asking the most audacious questions about fundamental life processes and provide them with great scientific environment, generous resources, and the last valuable commodity, time. Time to develop their ambitious programs, and inevitably, the new understandings that emerge from these laboratories bear upon the fundamental processes that underlie normal life processes and how these are altered in human disease. Rockefeller's breakthroughs are legion. They include the discovery that DNA is the stuff of heredity, the discovery of the hormone leptin, which is made by fat cells and regulates the appetite and activity via receptors in the brain, the discovery of dendritic cells, which regulate the immune response to infectious agents and cancers, and the development of combination therapy that transformed HIV infection from a rapidly fatal disease to one with a normal lifespan today. As one metric of the success of our scientists, there have been a remarkable 25 Nobel laureates in medicine and chemistry and 23 Lasker Award winners at Rockefeller since our founding. While this institution has a venerable history, the quality and impact of our science has never been greater. Rockefeller scientists have won more Nobel Prizes in medicine in the last 20 years than any other institute worldwide. And our faculty have received 1 Nobel Prize and 2 Lasker Awards in just the last 2 years. In September 2016, Charlie Rice received the Lasker Award for his pivotal contributions, leading to the development of a cure for hepatitis C, a viral infection that affects 170 million people worldwide and kills more than 350,000 people annually. Last fall, Mike Young was awarded the Nobel Prize in Medicine for his discovery of the molecular mechanisms underlying circadian rhythm, the process by which our sleep-wake cycle, feeding behavior, and metabolism is regulated and coordinated with the Earth's 24-hour day-night cycle. And just last month, David Alice received the 2018 Lasker Award for his wholly unexpected discovery that chemical modification of histone proteins, which form the ball around which DNA in our chromosomes is wrapped, plays a key role in regulating the expression of the genes encoded in DNA. Mutations that alter these histone modifications underlie diseases ranging from cancer to autism to congenital heart disease. And this pioneering work has spurred efforts to develop new therapeutics that target these histone-modifying enzymes, some of which are now in routine clinical use. Tonight, we will hear from 2 remarkable Rockefeller scientists whose work is transforming what we know about the proteins and protein complexes that are essential for life. We'll also hear about the revolutionary technology that is helping to drive their latest discoveries in structural biology, cryo-electron microscopy. Rod MacKinnon, our first speaker, embodies the Rockefeller ethos. Rod was at Harvard studying ion channels, proteins embedded in the cell membrane that allow selective passage in or out of cells of one ion, such as sodium, potassium, calcium, or chloride, but not others. These ion channels are critical to the life of every cell on the planet and also play specialized roles in humans. For example, sodium channels play a key role in in how neurons mediate the conduction of nerve impulses, which enable us to think, perceive touch and pain. Calcium fluxes in muscle cells activate muscle contractions that allow us to move at will. Potassium fluxes in the heart ensure the rhythmic beating of the heart. And when Rod started his own laboratory, the general mechanisms of how ions are conducted in and out of cells, as well as the specific mechanisms that permit this exquisite selectivity of ion channels, was completely unknown. And Rod thought that determining the atomic level structure of a channel would be necessary to address these critical questions. However, no one had ever succeeded in determining the structure of a membrane protein. And many experts in the field of structural biology thought that it was technically impossible or so unlikely to be successful as to not to be worth the effort. Rod was undaunted by these reservations and was recruited to Rockefeller to pursue this audacious ambition, and the rest is history. He succeeded in determining the structure of a potassium ion channel, which indeed illuminated the mechanisms of channel function. Rod received the Nobel Prize in Chemistry in 2003 for this pioneering research. Prior to being recruited to Rockefeller in 2014, Jue Chen, our second speaker, was a renowned structural biologist who had cracked the mystery of how a class of membrane proteins called ABC transporters, which use the chemical energy of ATP to import nutrients into cells. And she determined how the structure of these proteins and how they perform their functions. She did this in a brilliant series of experiments, making crystals and solving the structures of these transporters at distinct stages of the import process, getting a virtual time-lapse series of images of different stages of transport function. However, the function of the ATP exporters, transporters that use ATP to push certain molecules or ions out of cells, were almost entirely unknown. And this included the chloride channel called CFTR, which is the channel that is mutated in the genetic disease cystic fibrosis, a frequently fatal disease of children and young adults. Despite the discovery of CFTR in 1988, the recognition that CFTR allows chloride to leave cells, the mechanism by which this occurs remained obscure, and efforts to crystallize and solve the structure of CFTR, these efforts were unsuccessful despite the efforts of dozens of laboratories worldwide and undoubtedly several $100 million in funding. Rod and Jue's research in this period relied on methods that depend on first getting biological molecules to form highly ordered crystals. This allowed scientists to then shoot X-rays through the crystals to capture a two-dimensional diffraction image that could be interpreted to reveal the positions of all of the atoms in a molecule. The rate-limiting step in X-ray crystallography has been the crystallization step itself, which essentially entails try every condition you can possibly think of to get the protein to crystallize and hope for the best. This was the well-known dark art of crystallography, and many projects succeeded or failed based on the ability to get molecules to crystallize. And then just a few years ago, the field of structural biology underwent a technological revolution with the reduction to practice of a method that made it possible to determine atomic level structures without the challenging crystallization step. This method was cryo-electron microscopy, an idea that had been conceived many years earlier, but which took decades of technological improvements until the method suddenly crossed the threshold of being able to produce high-resolution structures without any need for crystallization. This suddenly opened a range of fresh possibilities to take on the most challenging problems in structural biology. Rod and Jue had both foreseen this coming cryo-EM revolution and took it upon themselves to learn the technology in its infancy. And convinced that the technology was ready for application, they persuaded the university to make an inspired investment, establishing the Evelyn Groos Lipper Cryo-Electron Microscopy Resource Center here at Rockefeller in November 2014. The center was created to meet the expanding needs of the university's structural biologists, and its instruments have been running nonstop 7 days a week ever since. The productivity of this group has been nothing short of extraordinary, with a trove of important papers published in Nature, Cell, and Science, the world's three most important science journals coming out of their laboratories. In addition to Jue Chen and Rod MacKinnon, Rockefeller's card-carrying structural biologists include Seth Darst, who studies the molecular machines that accomplish the reading out of genetic instructions, and Sebastian Klinge, who studies another group of nanomachines that are responsible for assembling ribosomes, the protein synthesizing machines present in every cell on Earth. Professor Tom Walls and assistant professors Greg Aleutian, both experts in cryo-electron microscopy applications, were subsequently recruited to Rockefeller over the last couple of years to take advantage of the emerging opportunities. And a growing number of investigators on our campus have been exploring the use of cryo-EM for diverse projects in cell biology, cancer research, neuroscience, and other fields. A great example of this is Vanessa Ruda, a brilliant young neuroscientist at Rockefeller who is studying the role of pheromones on behavior. The odorant receptors in insects have been very well studied, but their structure had never been determined owing to the inability to produce good refracting crystals. Vanessa recently used cryo-EM to solve the structure of the first member of the insect odorant receptor family, which was just published to considerable worldwide acclaim. It's safe to say this never would have happened without the technology and the expertise here on campus. Cryo-EM is now a global bioscientific phenomenon, and the three scientists who pioneered the innovations behind this revolution were recognized with the Nobel Prize in Chemistry in 2017. As the technology has transformed fundamental research in structural biology, it has also emerged as one of the most promising tools for drug discovery, potentially allowing the modeling of interactions of drugs or other molecules with their targets, with an eye toward altering drug structures in ways that can improve their effectiveness. Structural information about the moving parts of molecules can also inform the design of entirely new medicines. For academic institutions like Rockefeller, the technology's success has brought its challenges as well. The cryo-EM systems are incredibly expensive to acquire and very challenging to maintain. We could not begin to afford this technology without such generous benefactors as Evelyn Groos-Lipper, who's here with us tonight, Daniela Lipper-Cools, the EGL Foundation, Jim and Marilyn Simons, and Evan Greenberg. To meet the rapidly expanding demand of this technology on campus, we recently acquired our third state-of-the-art cryo-EM system. It's now being installed by Mark Ibrahim, the senior staff scientist who runs the Lipper Center. Prior to joining Rockefeller, Mark was employed by FEI, the manufacturer of these instruments, and we've been incredibly fortunate to have Mark here with us because his expertise has been central to getting these machines running at such a high level and with so little downtime. And Mark is here with us in the audience tonight. Now it's my privilege to introduce our speakers, who I've already told you much about. Rod MacKinnon is the John D. Rockefeller Professor and head of the Laboratory of Neurobiology and Biophysics. He joined Rockefeller from Harvard University in 1996, and he is the world leader in understanding the mechanisms of ion channels. Rod is an investigator of the Howard Hughes Medical Institute and an elected member of the National Academy of Sciences. In addition to the Nobel Prize, his many honors include the Albert Lasker Basic Medical Research Award and the Canada Gairdner Foundation International Award. Jue Chen is the William E. Ford Professor and was recruited to Rockefeller in 2014 from Purdue University. Jue is the world authority on ABC transporters that pump various types of molecules in and out of cells. These transporters normally play beneficial roles, but they can contribute to cancer chemotherapy resistance and are critical to understand. We'll hear about some of her amazing work this evening, including her determination of the structure and mechanism of CFTR, the cystic fibrosis protein, and how drugs that correct mutant function achieve their somewhat miraculous effects to help patients. I'm sure you will enjoy tonight's presentations, and now please join me in welcoming our first speaker, Rod MacKinnon, to the podium.
Thank you. Thanks very much, and thank you all for coming. So I'm going to talk to you about structural biology. And I'll start by saying the shape of things is essential to life. And I think that's clear to everybody, because if you look at things that we can see with our unaided eye, we see that plants that have to collect light from the sun have shapes and forms with branches and leaves and a broad surface area for doing that. Animals that have to forage and feed have shapes that let them move and do that. So it's clear that we say that the shape of something is very connected to its function. And this is true not only in what we can see with the unaided eye, but it's true when you look down in the microscopic world. So if you look at cells, you discover that in fact they have special shapes. So here on the left, so these are drawings, famous drawings from Ramon Cajal, neuroscientist, of neurons. And you can see that the neurons have very extended shapes, and that's because they're communicating with each other. One neuron passes the signal to the next. So what you see is a cell body with an extension in one direction and an extension in another. They're equipped with wiring. Here are pictures from a scientist I'll tell you about in a minute. These are of animal sperm. Sperm are cells. They're equipped with a tail because it has to swim to find an egg. Here are pictures of muscle cells. Muscle cells are spindly fibers with contractile elements inside them. And these cells are attached to each other so that they can contract and do mechanical work. So we see at the cellular level that shape follows form. And now if you go down even further, below the cell level to the molecules that make cells, we see there too that the shape of the molecules follows their function. And I'll show you an example of that. Let's consider an antibody, the protein in our immune system that's circulating. So when we are exposed to a foreign intruder, for example, a virus, an antibody neutralizes that virus by recognizing it. And the reason it recognizes it is because here on one surface of the antibody shown, it has a shape that almost perfectly matches a surface on the antigen. For example, a protein on the virus coat. So when those come together, they match like a key in a lock. This discovery, this principle of matching in the way antibodies works, was actually a discovery here at Rockefeller by Gerald Edelman. And it's a very beautiful example at the molecular level of why shape matters and why it follows function. Structural biology is the study of shape to understand how things work. And I'll tell you an example, just one example of an ongoing current project in my lab. So we don't know everything about it yet, but it's something I'm quite intrigued with right now. And then Dr. Chen will follow and show you some examples from her lab. But before I get to my example, I want to give you a little history of structural biology. And I would say, at least in my mind, structural biology of small things began with this man, Anton van Leeuwenhoek, a Dutchman who he didn't invent the microscope, but the microscope was shortly before his time invented, and he advanced it. And this is a theme you'll see in this little history, that it's technological advances that make science go in leaps and bounds. And in his case, he made lenses that were better than anybody else could make. In fact, better than anybody else could make for 150 years. His microscope is right here. There's a picture of him holding the little microscope up to his eye. The microscope involves a single lens and a pinhole. And he looked at things and he drew pictures. So this is his recording device, of course, his brain going down to his arm. And here he's drawing what he's seeing. His pictures are remarkable. So here is a picture of red blood cells that he discovered, what they look like. And here's a modern day picture of red blood cells. Pretty good, right? Showing the biconcave disc of red blood cells. Here are pictures of things he saw stuck to your teeth. things growing there, dental bacteria. And here are modern day pictures, and you actually can connect things to what he saw and drew. So light microscopy was really the first, first approach to microscopic structural biology. Now the light microscope, has been developed to be much better than what von Leeuwenhoek could use, but its limit had a fundamental limitation connected to the wavelength of light. Basically, you can't form an image of something that is smaller than half the wavelength of visible light, which is a limit of about 200 nanometers, a nanometer being a billionth of a meter. But practically, what does that mean? With a light microscope, you could see cells, you could see microbes, bacteria, you could even see some parts of cells, some things that were inside the cell, the larger things, but you couldn't go beyond that. Then in 1930, a new technology was invented, and this was called the electron microscope. And the electron microscope, very early on, started to get applied to biology. And one of the greatest successful applications of the electron microscope to biology happened right here at Rockefeller University. This is a picture from George Palati's work. A picture of a cell. This kind of cell is called a B cell. It's the immune cell, the kind that makes an antibody. And so here is the cell, and look what he could see inside. It's a very beautiful picture. You can see all kinds of organization. And in fact, these little black dots that are so prominent in here, these little black dots are actually molecules. They're large proteins called ribosomes. And for for this discovery and some other components of the cell, George Palati, along with two other Rockefeller scientists, Albert Claude and Christian Deduve, won the Nobel Prize in Physiology and Medicine. So actually, people say now that through the electron microscope at Rockefeller University, a new science was born called cell biology. Now, electron microscopes, although you could see the largest proteins as a black dot, you couldn't see any detail in it. And in fact, it wasn't until just a few years ago, and I'll tell you about that, that in fact, this was about as good as you could do with an electron microscope. You couldn't go further. It wasn't because the wavelength, it was for other technical reasons. So how to push down to get to the molecular scale? Well, that's a technique that you heard about from Rick Lifton. And there is a technique that has been used for over 70 years. And in fact, until just 5 years ago, was the technique of choice for molecular structure, and that was called X-ray crystallography. And you heard about that. It's a very good technique, but it's an extremely difficult technique because in order to get the structure, not only do you have to isolate your protein, but you have to crystallize your protein. And to do that is difficult with most proteins and impossible with many, especially the kind that Dr. Chen and I work on that are membrane proteins. They're notoriously difficult to crystallize. So that's the technique we used in our careers up until just a few years ago. But then 5 years ago, a technological breakthrough happened. And that breakthrough was the discovery of a new kind of electron detector. So the electron microscope was essentially the same. Actually, other improvements happened there. But this new kind of detector, which actually is the same in a fundamental way, it's funny to realize this, but it's fundamentally the same kind of technology in the photo detector in your cell phone camera. And actually, for a long time, people thought high-energy electrons would destroy it, but actually that wasn't the case. So this new kind of technology that improved what the signal to noise that you could get with an electron microscope, and it allowed the determination of a structure of a protein without crystallizing it, just by taking pictures of it in solution. So this was a real game changer. And when we realized this at Rockefeller, we turned to the administration and said, something big is happening in structural biology. And immediately, the university responded. And in fact, with enormous thanks to one of our trustees, Evelyn Lipper, we quickly established a cryo-EM facility with 2 very good electron microscopes. And in almost no time, this was considered one of the premier top cryo-EM facilities in the world. In fact, in terms of, for the first few years, I can say that in terms of scientific discovery, this was unmatched. So, in fact, we had 2 microscopes, and we're having a 3rd one installed right now into the facility, and here it is. It's hopefully, I think it will be ready to use very soon. But it's interesting to show this. I thought it was kind of funny to actually show this in contrast to a copy of the microscope that was used by van Leeuwenhoek, a picture of the microscope used by George Palade here, the electron microscope, and these are not even to scale. I mean, this is huge. And what's very interesting to think about this is, isn't it funny that in order to look at smaller and smaller things, we have to use bigger and bigger pieces of equipment and way more expensive. Let's see, yes, so now, what I want to do with the remaining, how much time do I have? I think, somebody say something. 10 minutes, perfect. Good. So now I'll come back to this. So that was history. I want to just come back in the final 10 minutes that I have to tell you about a project in the lab. And this project has to do, so here's the question. The question is, how do we feel touch? So I touch a place on my skin like this, or touch it with something, and I feel it. Right? How do we feel that? You probably think, well, what do you mean, how do I feel that? What I mean is actually something, of course, when I touched, we know that actually an electrical impulse or a series of electrical impulses traveled up the nerves from the back of my hand, went to my brain, and they came into my brain, and my brain interpreted this, I feel touch there. And in fact, the part about the electrical impulse going up the nerve, we actually understand that pretty well. That has to do with ion channels, and that's my area of research, and we have a pretty good understanding of how that impulse travels. What I'm talking about is, how did it start? What made it to begin with? By the way, you might be wondering, we have no idea how the brain said, I feel touch there. That's a question for another day. It's a very difficult question, how the brain interprets things. But I'm talking about the very simple question. How did the electrical impulse start when I touched? Okay, and so I mentioned that electrical impulses happen by ion channels. And here I drew a little cartoon to explain in a rough sense how this works. So here is a cell, my drawing of a cell, nucleus and some endoplasmic reticulum, a mitochondrion, and it's surrounded by a membrane. And here I draw a little circle and a little piece of membrane, and I blow it way up here in a way that you can't really do, but we're looking at an experiment in our mind. And these things are lipid molecules that make this thin membrane that surrounds the whole cell. And in this membrane, there's a protein called an ion channel. There are many of them throughout a cell. When that ion channel opens, ions flow across it. And because ions carry a charge, when they flow across the membrane, they make a charge difference, which I indicate by putting a row of pluses here and minuses here, because plus is running in, making it more positive on the inside than the outside in the picture I've drawn. And that charging of the membrane makes what we call a voltage difference across the membrane that can be measured. And in fact, if an ion channel opens, the voltage changes there, and that effect propagates across the cell membrane, very much like a voltage change propagates in a copper wire conducting electrons. And that's, in the simplest way I could say it, the essence of how an electrical impulse travels across a cell membrane. And if that cell membrane is a neuron with a long extension, it will travel up that extension like a wire. So where does it start? So 8 years ago, scientists at the Scripps Institute discovered a gene called the piezo gene. that makes an ion channel that actually has a property that it responds to mechanical force. No one knew what it looked like, but it had that property. And here's what it looks like in an experiment. If you take a cell that has this kind of ion channel in it, and so this is a recording, and here we're plotting membrane voltage. So that's a measure of the charge difference across the membrane of that cell. And now here, this is indicating, and this is time, and here at this time, that little square is just marking a time when we touch the cell and then stop touching it. So all they're doing is touching the surface of this cell. And what's quite striking is when you touch the surface of this cell, the voltage changes. And the reason that happened is because this piezo ion channel somehow felt the force of touch. When the membrane was touched, it opened up, let ions run, and changed the voltage across the membrane. So that's what we would need to understand how do I feel touch? What is the initial step? And so the question is, how does this work? How does a channel do this? So to try to answer this, we'll come back to function and form, or shape and function. What can we tell if we could see this piezo channel? Would it tell us how it's working? Now, just before I show you the piezo channel, I'm going to show you some other ion channels, some other typical ion channels. And here they are. There are 3 different ones. I won't tell you what ions they conduct. That's not important. They have a hole. Each of them have a hole down the middle. So this gray thing is the membrane. And I'm showing the outside of the cell would be on the top, and inside the cell would be on the bottom. So here's a gray membrane and 3 channels sitting in there. And one thing you notice about these channels, okay, I tell you there's a hole down the middle because it's true. But one thing you notice about it is that the part in the membrane is made of these structural units called alpha helices, and they're bundled together into like a flat disk. You see what I mean? This guy's a flat disk. This guy's a flat disk. This one's a flat disk. And that's because they sit nice in a flat membrane. So that's what all ion channels that we've ever seen look like. And in fact, that's what all membrane proteins that we ever saw look like. So now let me show you piezo. So we used the electron microscope, and we got a structure of this thing to try to figure out how it works. Here it is, okay? It's very different. So it has a pore. So this is the outside the cell. This is the inside the cell. It does have a pore down the middle, like the other ones. In this case, it's closed. And these helices, of course, these are the kind you saw in the other channel proteins that went back and forth across the membrane. But the weird thing is, it's not flat. It's all bent up. You see that? It's all curved up. Now, so in a membrane, it would have to be like this. It would have to bend the membrane. In fact, not only would it have to, but it does, because we take pictures of it inside membranes, and this is what it does. It actually bends the other. This looks like 2 membranes, but the representation is just the 2 sides, so you can see. So in that otherwise flat membrane, where the piezo is, it bends it into what we call a dome or a cap. Okay, so why does this happen? What does this mean? So to explain this, now I have to tell you one concept from physics that if you understand, you'll look at this and say, wow, it will speak to you. Okay, and the concept is this. So I'll take it in 2 steps. I think you probably all know this. If I want to know how much work it takes or how much energy I have to put in it to lift up a weight, the way I calculate that is I know what the force is, because I know its mass times the gravitational acceleration is the force. It's being pushed down to the floor. So that's the force. And I lift it up a certain height, and it's the force times the height is the amount of energy. That's the energy. I lift up twice as high, I have to do twice as much energy. So that's a one-dimensional problem. I'm just lifting it up in height. This thing is sitting in 2 dimensions. And so if you translate that force times distance equation into 2 dimensions, it actually comes out as something called tension times area. So tension is like the force. It's actually the force per length times the area is the work you have to do. And what's that mean? That I'll tell you in a minute. But what that means is if there is some way that piezo could expand its area, and it's the area in the plane of the membrane, that would be the work difference between this shape and another shape. And explicitly, I'll say that imagine piezo could flatten out. And it turns out if you look inside this structure in detail, you realize that if you flattened this bowl, the pore would open. It's rigged such that that would happen. So if it flattens, the pore would open. And what flattening is doing here is actually, so here's another little hand-drawn cartoon, is the closed shape, little representation of that, with a closed pore, very narrow. And here is an imaginary flattened one with an open pore. What happens when I flatten this area under it, that is the area projected into the plane, you see gets much bigger. And so if you put a tension in the membrane, which is going to be a force that pulls out, which is, for example, what happens when we push on the cell membrane to kind of stretch it, pull it taut, put tension in it. Because the piezo channel has this cup shape, it can go pop and open. If it did not start with this cup shape, it couldn't do that, because the energy is related to the force per length, called tension, times the area difference between this one and this one, which is a big positive value, which is to say the piezo is like a spring, ready to pop open. And when it pops open, its area becomes wider. And that makes it exquisitely sensitive to a tension on the cell membrane. So then coming back to what I began with, and that is looking at the form or the shape of something and connecting it to the function it does. If you know the physical principle I just described, then you look at this protein, and I would say, I see maybe some as good, but no more beautiful example of evolution leading to a structure or functional or shape that is directly connected to the function that this protein does, which is to sense tension, which is, I think, why when I touch, some of those channels in the nerve ending are going pop. Ions rush in, and it initiates the nervous impulse that propagates up. Okay, I'll stop there, and I probably went over.
So today I'll tell you two stories. The first story is a scientific quest to understand how come certain tumor cells are so hard to kill. Chemotherapy is basically using the chemical reagent to kill tumor cells, and has been implemented for more than half a decade with mixed success. Some complications of chemotherapy is because some forms of tumors, when they first respond positively to a drug, will come back in a form it becomes resistant to this drug. Not only the drug it has been treated with, but also a variety of different drugs. And those tumors are becoming impossible to kill by chemotherapy. So to understand what makes those particular tumor cells resistant to so many different drugs, a phenomenon we call multi-drug resistance, many scientists ask this question in their different ways. So early on, in 1973, a Danish scientist called Daniel, actually he was assistant professor at Rockefeller at that time, recognized the ability of tumor cells to reject drugs are dependent on energy. So most of the calories we take in every day are turned into a form called ATP that store the energy and the ATP will go around our body to power different machineries to enable us to do what we do. And what Daniel found out is if you deplete those tumor cells from ATP, they're no longer resistant to drugs. So he says, maybe those tumor cells also have this molecular pumps that's powered by ATP to pump different drugs out. That idea was not very popular at the time because it's very hard for us to imagine a particular pump can recognize so many different drugs. Usually one molecular pump recognizes one kind of drugs. But in this case, if it's a drug pump, it has to be able to recognize so many different drugs. So most of the people believe at that time, actually it's not about the molecular pump, it's perhaps something happened to those membranes of those cells. Those membranes becoming impermeable to drugs, so nothing can get in. So that's why 3 years later, when 2 Canadian scientists finally isolated a kind of protein that's only found in drug-resistant cells, not in normal cells. So they say, hey, those must be the protein responsible for drug resistance. And they named this protein P-glycoprotein. P stands for permeability. So what they believe is this protein makes the membrane impermeable to drugs. And meanwhile, American scientists led by Michael Gottesman has identified the gene that's responsible for drug resistance. So he named the gene MDR gene, so multi-drug resistant gene. So what he identified is if you knock out this gene, the cells can no longer resist drugs. So we all know genes are blueprints to make protein. So the question is this MDR gene make the P-glycoprotein? Luckily, scientists can work together. So Victor Lin and Michael Gottesman start to work together, collaborating, and then they showed us, indeed, the MDR gene makes the protein called the P-glycoprotein. And it turns out, Daniel was right all along. P-glycoprotein is a pump. It belongs to a group of pumps we call ABC transporters. So ABC transporters are everywhere in all kinds of life, bacteria, plants, human. What they do is they use the energy stored in ATP. They'll burn ATP into ADP and phosphate and use this energy to either bring nutrients into the cell to support the cell growth or pump out toxic molecules, hormones, or any molecules it needs to pump out to communicate with other cells. So we do not make P-glycoprotein to resist drugs. They are here for our benefit. So normally, P-glycoprotein functions to protect our important organs. For example, in the brain, when blood streams meets the central nerve system, they're lined with those cells expressing P-glycoprotein. So P-glycoprotein are there to prevent toxic molecules to enter our nerve system. Also, they're found in the membranes to protect our unborn babies. Unfortunately, many drugs we use to treat all different kinds of diseases are also being recognized by P-glycoprotein as toxic molecules. So this is just only a partial list here. So for us to understand how P-glycoprotein works, first I want to convince myself this molecule is responsible for drug resistance. So I'm just going to show you data from a very simple experiment. So we take cells we grow in the lab that normally do not express P-glycoprotein. So in this case, those are insect cells. Then when we grow the cells in the presence of Toxo, which is a very common anti-tumor drug we use, at 0.1 micromolar, you can see the cells lose their shape, they actually die. So we take the same cells and now make them to express P-glycoprotein, and we tag the P-glycoprotein with some green fluorescence protein so we can know what we're looking at are indeed the cells express P-glycoprotein. Now you can see the cells grow happily, even at the highest taxol concentration we test. So imagine this is the tumor cell. In this case, Taxol can simply not get into the, kill those cells. And we can actually quantify the number of cells can survive. With normal cells without P-glycoprotein, 0.01 micromolar Taxol will kill half of them. But when P-glycoprotein is expressed, even at the highest concentration without the drug crashing out, we just cannot kill the cells. So to understand how P-glycoprotein works, how does it pump Taxol out? We look at the drug, we look at the structure. So there's a lot to see, but I just want to focus on two parts. One part is inside the cell, those two globular domains are responsible to bind and hydrolyze ATP. So if you imagine ATP is the gas, those is the engine part of the pump. In this particular conformation, those two engines, those two ATP binding domains are separate from each other. Another part I want to discuss is this part of the protein that inserts into the membrane. And in the middle of this pathway, they have a pocket suitable to bind the drug. And to help you seeing that, I outlined the pocket with a green mesh. So this is inside the membrane, a pocket formed by the protein that will bind many different kinds of drugs. But there's something very interesting about this pocket. This pocket is open towards the membrane. Normally, transporters will work, a binding pocket is opened inside the cell or outside the cell, but P-glycoprotein is different. It has this pocket open into the membrane. And this opening is telling us a lot about how P-glycoprotein works. When we treat cells with drugs like Taxol, first thing they need to do is to go through the membrane to enter the cell. And in this case, Taxol will find molecules that will block cell division. But because the pump has a binding site for Taxol and also opens to Taxol in the membrane, what they can do is actually suck the drug from the membrane before it even reaches inside the cell. So when the drugs are diffused into the membrane, it will bind to P-glycoprotein and stay inside the pocket. So what happens next? How does the drug get transported out? Remember, this is a pump need ATP, so this is what ATP will do. It turns out when ATP is present, the protein completely changes shape, and it looks very different now. The two ATP binding domains now come together, and when they come together at the interface, they have two ATP molecules bind right there. So those ATP molecules are like a glue to glue the two halves of the pump together. The cavity I showed you before now is completely shut off from the membrane, from inside the cell. It's now facing outside. It's actually collapsed in a way the drug has to be pumped out, squeezed out. So another property about this shape is when ATP is positioned here in between the two domains, it's ready to be hydrolyzed. It's not ready to be burned. So in this conformation, ATP will be hydrolyzed, and hydrolyzing ATP will actually destabilize conformation and make it back to the open state. So here I just want to put it together. This is P-glycoprotein without ATP. The two engines are separated and the cavity is open to the membrane. And when ATP binds, it comes together and squeezes the drug outside the cell. So we just summarize what we learned in this little animated movie. Drugs like Taxol will have to penetrate through the membrane to enter the cell. But because the pump has an opening for the drug, it functions like what we call a hydrophobic vacuum pump. It sucks the drug into the protein. At the same time, ATP will bind and reshape the protein. Now the drug is squeezed out of the membrane outside the cell. At the same time, these two ATP molecules will be hydrolyzed, and afterwards that energy will be used to reset the pump to its original conformation. Now it can pick up another drug and it keeps pumping substrate out. So the second part of my talk, the second story I'm going to tell you is about a boy named Louis Wilker. Louis lives in England. He's 8 years old. He's becoming quite well known nowadays because of the letters he wrote. He wrote letters to the leaders of England and to the leaders of Vertex, a drug based in Boston. He wrote these letters because he needs help. He is quite sick. He suffers from a disease called cystic fibrosis. So he has to take medicines to help him breathe. And also he gets bacterial infection quite often, so he needs to be on antibiotics quite often. And what's going on with Louis is cystic fibrosis. One of his genes called the CFTR, located on chromosome 7, has mutations. It's not a normal gene. All of us have 2 copies of CFTR gene, one from each parent. We only need the one good gene, CFTR gene, to be healthy. But unfortunately, both copies of Louis' genes are defective. So he cannot make a good protein of CFTR. So Louis is not alone. Actually, cystic fibrosis is quite a common disease. So Louis's parents each have a copy of a mutated CF gene. So they are called the carriers. Right now, I estimate 1 in 30 Americans are CF carriers. Their children have 25% of chance to get disease. And that that percentage doubles if one of the parents actually have cystic fibrosis. So the CFTR gene encodes an ion channel that's responsible to let chloride ions go in and out of the cell. Because chloride ions are in a delicate balance with water and the pH value, so a proper function of CFTR channel is required to make the mucus outside the cell to be watery and slippery. When something goes wrong with CFTR protein, what happens is mucus will build up outside the cell, and those mucus will cause a number of problems in our body. So in the lung, the mucus will make difficulties to breathe, and also those mucus serves as a base camp for bacteria to grow. That's why patients get a frequent infection. In pancreas, the buildup of the mucus will block secretion of enzymes that's necessary for us to digest food. That's why children has to take medicine to help them get enough nutrients. And the male with CFTR are infertile because of blockage. So to understand how CFTR work, we and many, many others have tried for a long time to crystallize CFTR without success. So only until 2 years ago, with the ability to use cryo-EM microscopy, we were able to take a look at the structure of CFTR. At the first glance, you'll say, hey, it doesn't look that different from the drug pump I just showed you. That's not surprising because CFTR is also an ABC transporter. It shares a common ancestor with the P-glycoprotein we just shown. But the details in its structure make it a chloride channel, not a drug pump. Another difference is this novelty, this red domain I showed here that jams in between the 2 halves of the protein. And this is what we call the regulatory domain. This domain actually controls when the channel is turned on and off. So in this particular conformation, the regulatory domain is jammed in between the 2 halves, so the channel is short. And in a different conformation, which I'll show you here, after phosphorylation of the regulatory domain and ATP binding, CFTR looks quite different now. So this red domain now get out of the way. The two ATP binding domains, just like a P-glycoprotein, come together with two ATPs bind at the interface. And now the transmembrane region forms an open channel for chloride ion, and I'll show you here. So that's outlined in this black tube here. So different from the drug pump, the chloride pathway doesn't open to membranes. It opens inside the cell and outside the cell. That's because chloride ions do not enter the membrane. Also, this tube is lined with positively charged residues. We know chloride ions are negative charges. Negative charge and positive charge like each other. So those positive charge residues will function as a guide to attract chloride ions to go through this tube, to go in and flow in and out of the cell. So I mentioned many different mutations will cause cystic fibrosis, and the symptom of patients depends on the type of mutation. So about 5% of patients have mutations that they cannot make CFTR protein at all. And the majority of the patients can make CFTR, but some of the CFTR protein cannot fold well, which means it cannot form the shape I just showed you. And our cellular machinery is controlled. Only correct shaped molecules will make to the cell surface. So those patients will make CFTR protein, but they stuck inside the cell. And another portion of patients will also make CFTR protein, and they can fold correctly to get into the cell surface, but they will not conduct chloride as it should be. So one question we can ask is, where does this mutation look like? Where are they? How come a single amino acid change in those patients will make the protein not function? So here is a map of different mutations that cause cystic fibrosis mapped onto the structure of CFTR. Each one of them is shown as a little bar here. So I color them differently based on what they do. For example, those purple bars are the residues that outline that pathway for chloride. So change of those residues either collapse the channel for chloride or change the positive environment so chloride ions cannot flow smoothly. And other mutations, for example, are located in the ATP binding region. So this region, mutations here will interfere with the ability of CFTR to bind and hydrolyze ATP to form the right shape that will open the pore. And I want to just focus on one particular mutation that's found in about 70% of the patients, which is the phenylalanine mutation. In those patients, this particular residue is missing. So this residue is located at where the ATP binding domain, colored in blue here, in contact with the green helices that form part of the chloride channel. So if you consider the protein is made of pieces, then the ATP domains has to assemble into the chloride channel part to make sure they can communicate with each other. And phenylalanine 508 is like a screw at the interface to make this assembling properly. So patients without this residue, this missing an important connection, there's no wonder this blue domain can no longer attach to this part of the molecule, and then cells consider this molecule not folded, and those mutant CFTRs are stuck in the cell and they cannot reach to the cell surface. So what science has done in order to improve patients' life, we have made quite a difference. From in the early 40s, the patients usually cannot survive infancy, and with the discovery of treatment of enzymes to help digest food and to clear airway, antibodies to prevent frequent infection, and the lung transplant. With those treatments, the average survival age reached to about adulthood. And just about a few years ago, Vertex, a company, put on 2 kinds of drugs that will specifically activate CFTR protein. And with those drugs, the current survival age is 41 for patients, and we have to see what the long-term effect will be. So what are the issues here? There are several issues. All the treatments are treating cystic fibrosis, but not cure it. So, also, not every patient will respond to the drugs that's available. But a more emerging issue is the affordability. So this is just a general care including clearance of the airway and antibody treatment for a CF patient annually costs average about $50,000. So 50 years of treatment will cost about $2.5 million. The Vertex drugs right now in the US cost $270,000 per year, and the 50 years of treatment will be cost $13.5 million. Not everybody can afford this. In fact, many countries, including Canada, UK, the Public Health Service simply said, we cannot afford this drug, and we cannot have our patients have access to Vertex drugs. That's why Louis wrote those letters to say, this is addressed to Mrs. Hunt. She is the head of research at Vertex. In the letter, he said, you have the medicine that can make me feel much better and not have to spend so much time in hospital. Please sell it to my country. If your son had cystic fibrosis, I know you'll understand and lower the price of Okabe. Thank you, Louis Wilker, age 8. So the question I ask myself is, can we make a difference? So traditionally, those drugs are found by drug companies by screening hundreds, thousands, and millions of compounds. So they set up an assay, take cells that has defective CFTR. Then they treat each cell with different drugs they have, and then find the ones that can increase the activity of CFTR. And this is by screening 400,000 compounds. Vertex identified their first drug. And now it's been on the market for 5 years, but still no one knows where this drug binds, how they work on CFTR, why can they improve the activity, why they make patients feel better. So we think maybe that knowledge will be useful. What we did is take the Vertex drug and determined its structure with CFTR, and we find a pocket where the Vertex drug sit there. And by looking at how Vertex drug interact with CFTR, we understand now this drug can stabilize CFTR in the conformation. The pore will stay open longer, so the more chloride ions can go through. Then we look at another drug that's developed by Galapagos, a company in Belgium, and also determined its complex structure, its conformation with a bond structure with CFTR. And to our surprise, this drug, which is screened from a completely different library by a completely different company, bind to exactly the same position. Here we say, wait a minute. Now we see a spot on the CFTR protein where compounds bind there will make it more active. Maybe we can use this information to find more compounds can also make CFTR active, perhaps even with higher efficiency and more specific. So this is what we're currently pursuing now. We team up with a scientist named Brian Shortcat at UCSF. He's an expert in computational biology. So instead of screening every compound, millions of compounds experimentally, Brian took the structure we have and used the computer, asked the computer, say, tell me what kind of chemical moieties will bind to this pocket. So within a few months, he screened a few millions of compounds and find the 58 compounds that he predicts will bind to this pocket. So he made those compounds ship to my lab, and we test them one by one. And about 10 of them actually activate CFTR, and one of them is already in the range of those drugs that's on the market. But we're not stopping there. So the next step, what we'll do is we'll determine the structure of our compound and see how it interacts with CFTR. And from that picture, we might look at the structure and say, okay, now I see a pocket here. Maybe I can add another chemical moiety to make it bind stronger. And I see some positive charges here. Maybe I can put a negative charge so it can bind even stronger. So through iterations of what we call the medicinal chemistry and the structural biology, we hope one of our compounds can eventually develop into drug to treat cystic fibrosis. If that happens, I will make sure that drug will be affordable to everyone. Kids like Louis will not have to struggle for their chance for life. Thank you.
Okay, that was great. Maybe I'll ask a first question. Rod, you eloquently gave us the sweep of history of going from von Leeuwenhoek and his microscope to Palade and the electron microscope and to the beautiful presentations of structural biology of membrane proteins with cryo-EM. I think as scientists, we're really good at thinking about what we can do today, tomorrow, and maybe next year. But if you look at how radically technology has changed over these 100-year and 20-year periods, the pace of technological revolution is extraordinary. So if you look out the next 5 years, the next 20 years, the next 50 years, what's your crystal ball tell you are the next big transformations in this area?
Well, I'll answer it this way. I think that the revolution that is taking place right now because of advances in electron microscopy, it made a big jump, but it's actually, I would say, we're only on a steep incline. It didn't make a jump and level off yet. And the reason is there's a new method being developed in a number of labs and actually being done here by Greg Alushin already, and we're trying it in our lab, called tomography, where you're using the electron microscope, same detector, but in a way where you tilt the stage and you get pictures from different angles. And what this is going to ultimately allow is to get structures in the cellular environment. There are some challenges to be overcome, but imagine right now, everything you saw that I talked about and that Dr. Chen talked about, these were isolated proteins that we took from the cell. The next step, and I think we'll be able to do it with the technology at hand, we'll actually be able to see the protein structures in the cell, in the cellular structures, which means you'll be able to see the protein structures with their neighbors that they interact with. And I see that actually happening in the next 5 years.
That's very exciting.
This will be huge. And so I think this area is a watershed right now. And actually the new technology we're just beginning to see what it's going to allow us to see.
That's very exciting. Questions in the audience? We have a question over here and another back in the back row over there. If we could get a microphone there, we'll take this question first. Thank you.
Hi, so to simplify this, the cryo-EM allows you to see the shape and structure of a molecule that you can then synthesize and then apply as a drug to cure some shortcoming? Is that my understanding of what's happening here?
That's your question.
The cryo-EM enables
Yes.
you to see the structure of a molecule and the shape of a molecule that you can then recreate and apply as a drug application.
This is one example, yeah. So basically, this allows us to see more and more structures. We were able to see some structures of molecule before by using X-ray crystallography, but cryo-EM makes us to see those molecules previously we cannot crystallize. By seeing them and see how they change their forms by interacting with other proteins, or by a response to force, or by interacting with ATP, or by interacting with drugs, we understand better and better how molecules work. And because those molecules are important for our health, and this knowledge eventually will translate to developing drugs that will help us.
And this is just only 5 years old now, cryo-EM.
Cryo-EM for single molecule determination is only 5 years old, that's right.
Thank you.
Back in the back row.
So in your descriptions of the protein channels, you say that there are charged residues that are within the membrane. How can these charged residues exist so close to the hydrophobic cell membrane?
Excellent question. So what the question is referring to is cell membranes are greasy. So usually charged residues will not be happy there. They want to be in the water. But the CFTR is shaped in a way, those charged residues are actually aligning a pocket where it's normally filled with water. So that's what makes the charged residues happy. Excellent question.
Other questions? Yes? Where's there? There's a question here, a question there.
Hi, thanks very much for this really illuminating talks. I know the focus is on ion channels, but I was wondering if you could care to opine or speculate upon advances in determination of G-protein coupled receptor structure, about the diversity that may be known in some of the structures that have been solved through cryo-EM. And then on top of that, I was also curious to know, because there have been discussions about rational drug design, and soluble proteins are well known. How do you think the advances in knowledge of ion channels and G-protein coupled receptor structure will actually translate into better drug discovery efforts?
So I'll answer the first half part, which on the GPCRs, cryo-EM has made a huge change in terms of many structures now have been determined that were not determined before. So it's advancing that field, just like it's advancing almost every field of protein, where the protein is bigger than 100 kilodaltons. As for the drug-related question.
Yeah, so actually what I described for CFTR is exactly happening for GPCR also. There are many more structures of GPCR, how they interact with drugs are available. And I happen to know my collaborator, the same person who does CFTR computation, is also working on a number of GPCRs with the same idea, finding novel chemical moieties that can be drugs.
So just following up to your point, many of the known drug targets today are ion channels and G-protein coupled receptors. And in the case of the G-protein coupled receptors, one of the current trends in the field has been trying to identify so-called biased agonists. There are multiple paths downstream from the G-protein coupled receptors, and there have recently been developments that suggest that you might be able to separate out with selective molecules molecules that activate just one part of the downstream pathway and not others. And the structural biology, I think, will provide incredible insight into the kinds of molecules that are capable of doing that. And I think holds a lot of promise for getting a lot more mileage out of targets that we've known about for a long time and being able to differentiate some positive effects from some negative effects that are attributable to current drugs, and dial the bad ones out and emphasize the good ones. A question here?
This is fascinating. Let's turn to economics for a minute. If you're able to bring that little boy's cost from $270,000 down to something reasonable, that's a good thing. And I think you ought to be able to share in that economic gain. And our friends across the street at Sloan Kettering ran into some difficulties as it related to how to share it. And I think you all ought to share, but maybe the president might be able to speak to how you manage that very complex process where you have to work closely with drug companies. You mentioned it a number of times, but there's an economic stake that's quite large. And how do you handle all that?
Do you want to start?
You want the real answer? I have no idea. I just feel, as a scientist, we got to be able to help. And my goal is not to make money. I'm working on this because I think everyone should be able to have access to those drugs if they need to. But practically, I have no idea how to translate the pictures I show you into a drug. I will need help.
Addressing your point directly, I think there are several elements that are germane. The first one is competition. As long as you've got a monopoly on a drug, you can charge whatever price you want currently, right? We don't tell drug companies what they can charge for drug pricing. And the only market forces that push against them are places like Canada and UK that say, well, we're not going to allow those drugs to be sold for reimbursement by the Public Health Service in Canada or the UK. That's the only game in town. But if you've got multiple drugs that can do ostensibly similar things, some might be a little bit better, some might be a little bit worse, but both have significant efficacy, then there's real pressure on price, because in order to get market share, a new drug coming in would necessarily come in at a lower price point, and then there would be a response that would work its way down from there. And we have a practical example of that. I mentioned Charlie Rice's work toward the development of the drugs for hepatitis C to cure this chronic viral infection. When those drugs first hit the market, they were listed at a price of about $95,000 for a curative regimen of therapy. As soon as a second company came in with their own version of same approach to a cure, the price immediately was cut in half, and the prices have been coming down from there subsequently. So drug pricing is clearly a complicated issue, but competition is not a bad way to try to foster more realistic pricing. The other part that gets even more complicated into the equation is that drug prices are in part determined by how long a company has exclusive rights to the patent on that molecule. And that's currently, I think, 17 years. So from the time you patent a drug to the time it comes to market and can be sold for reimbursement, companies typically find that they've got 7 to 10 years left on the patent life in order to make back and recoup their investment, plus have money left over to do the development of the next transformative therapy. So there's pressure during that window. And of course, many of the drugs that we take now that were expensive when they first came to market are now available for $4 a month at Walmart. So once they come off patent protection, they can become very cheap if the drugs are inexpensive to synthesize and distribute. So drug pricing is clearly complex, but I'm very optimistic that not only is there a route to creating a competitive molecule, but it might even be a better molecule, because up to now, nobody has had any idea how to modify a molecule that works as well as the existing Vertex drugs. But now that you can see exactly how the drug is binding at the atomic level, as Jue Chen said, you can think, where could I add a side chain to that molecule that might make the binding stronger and improve the function of the channel? And I think this is a revolutionary technology for achieving this.
Can Rockefeller University benefit in some way from that great economic accomplishment?
Surely, and we have a technology transfer office that does file patent protection for novel discoveries that have utility as a drug might. But we would still, we are not going to get into the business of making the drugs and distributing them ourselves. We would eventually be licensing the intellectual property to a company that would then be responsible for going through the clinical trials and developing the molecules for therapeutics. So the equation still remains somewhat complicated, but we certainly have leverage as to the terms under which we would license a molecule going forward.
Then you give her a raise.
She's already deserved that. That's not the question.
As you can see, I'm in very good hands.
But actually, she really means this. Look at what it says under
That's why
Rockefeller
we're
University.
here.
That's why we're here.
No question. Thank you. We have time for another question back
sorry.
over here. And if
Sorry, this is my last question, but I'm really fascinated by the topics. You mentioned, one area is to be able to see the membrane proteins in the cell, right? I was just curious to know other, parameters for technological improvement. Maybe you can comment on dynamics, molecular dynamics, and other parameters.
Yeah, that's a very good question. So the question being asked is, I'll rephrase it, and that is, even if you can get structures of entire cells, and there are still great challenges to get that, the structures would be static pictures, but we know that really cells, everything's dynamic and interacting and moving, and there is a whole kinetic process of life, and it's a very good question, and in fact, that's sort of another kind of branch of molecular research that's going on that has to do with looking at interactions in real time. And while you can't do that at the same kind of resolution that we can see molecules using the technique, the cryo-electron microscopy, there are ways now, there have been fantastic light microscopic imaging methods where you label the proteins. So while you can't see the protein in detail, you can see precisely where the protein is in the cell at very good resolution. And you put multiple labels on it, and you are targeting proteins that interact with each other in a signaling pathway. And then you can create a movie of how that's happening. And that's another big, important area, in fact, that part of my lab works on. And many, many scientists do. And because you're right, it's not just a static picture.
Well, I'd like to thank Rod and Jue Chen.